Very good morning, students. Today we are going to discuss the primary structure of dicotyledonous stem. We are all very familiar with the levels of cellular organization. Atoms group to form molecules, molecule into organelle, organelle into cell, cell group to form tissue, tissue group to form tissue system, tissue system group to form organ, and organ into organ system into organism. Let me see the primary structure of dicot stem. See the picture, morphology of a dicot stem. Morphology is nothing but the external feature of dicot stem. So externally we can see terminal bud, bud scales, axillary bud, then nodes, internodes, leaves care. What about the internal structure of dicot stem? So the primary structure of dicot stem, for example sunflower stem, reveals epidermis, cortex and stele. So it comprises three different types of tissue systems: epidermal tissue system, ground tissue system and vascular tissue system. This is the ground plan of internal structure of dicot stem. Here we can see epidermis with epidermal hair, cortex with hypodermis and endodermis, then vascular bundle and pith. Let me see the primary structure of dicotyledonous stem, sunflower stem, in detail. So this is the transverse section of dicot stem. Internal structure of dicotyledonous stem reveals epidermis, cortex and stele from the periphery towards the center. Epidermis. Epidermis is protective in function and forms the outermost layer of the stem. It is a single layer of parenchymatous rectangular cells. The cells are compactly arranged without intercellular spaces. The outer walls of the epidermal cells have a layer called cuticle. The cuticle checks the transpiration. The cuticle is made up of a waxy substance known as cutin. Stomata may be present here and there. Epidermal cells are living. Chloroplasts are usually absent. A large number of multicellular hairs occur on the epidermis. So epidermal tissue system includes epidermis, cuticle, stomata and epidermal hairs. Here it is multicellular hairs. The major function of epidermis: protection. Cuticle checks transpiration. What is transpiration? Loss of water in the form of vapor through aerial part of the plant body. That is called transpiration. Then stomata also play a prominent role in transpiration and gaseous exchange. Next, multicellular hairs. Next to epidermis, we can see cortex. Cortex lies below the epidermis. The cortex is differentiated into three zones in the case of dicot stem. Below the epidermis, there are a few layers of collenchyma cells. Just see the picture. Below the epidermis, there are a few layers of collenchyma cells. This zone is called hypodermis. Hypo, below. Dermis is nothing but the epidermis. The tissue which is present below the epidermis called hypodermis. What is the major function of hypodermis? It gives mechanical strength to the stem. These cells are living and thicken at the corners. Inner to the hypodermis, a few layers of chlorenchyma cells are present with conspicuous intercellular spaces. This region performs photosynthesis due to the presence of chloroplast. Some resin ducts also occur here. Next, third zone. The third zone is made up of parenchyma cells. These cells store food material. So the major function of parenchyma, storage. So these are all the three different zones in cortex of dicot stem. The innermost layer of the cortex is called endodermis. The cells of this layer are barrel shaped and arranged compactly without intercellular spaces. Endodermis is also called as starch sheath. The starch grains are abundant in these cells, so this layer is also known as starch sheath. This layer is morphologically homologous to the endodermis found in the root. In most of the dicot stem, endodermis with Casparian strips is not developed. In root, we can see Casparian strips and passage cell in the endodermis, but in dicot stem, endodermis with Casparian strips is not developed. Next, stele. So the central part of the stem inner to the endodermis is known as stele. What is stele? It consists of pericycle, vascular bundles and pith. In dicot stem, vascular bundles are arranged in a ring around the pith. This type of stele is called eustele. So the outermost layer of stele is known as pericycle. Next to stele, that is next to endodermis, we can see pericycle. This is the outermost layer of stele. Pericycle is a layer of cells that occur between the endodermis and vascular bundles. In the stem of sunflower, a few layers of sclerenchyma cells occur in patches outside the phloem in each vascular bundle. See the picture. The vascular bundle consists of xylem and phloem. Just above the phloem, we can see a patch of sclerenchyma cells. So a few layers of sclerenchyma cells occur in patches outside the phloem in each vascular bundle. This patch of sclerenchyma cells is called bundle cap or hard bast. The bundle caps and the parenchyma cells between them constitute the pericycle in the stem of sunflower. Next, vascular bundle. The vascular bundles consist of xylem, phloem, cambium. Xylem and phloem in the stem occur together and form the vascular bundles. These vascular bundles are wedge shaped. So in dicot stem, vascular bundles are wedge shaped. They are arranged in the form of ring. Each vascular bundle is conjoined, collateral, open and indark. So what do you mean by conjoined? Xylem and phloem arranged in the same radius. Collateral, xylem and phloem arranged in the same radius, phloem facing outside. This is called collateral. Open, cambium is present in between the xylem and phloem, then the bundle is called open vascular bundle. Endark, protoxylem points facing towards the center and metaxylem facing towards the periphery. So this condition is known as endark. So in dicot stem, vascular bundle is conjoined, collateral, open and endark. Next, phloem. Primary phloem lies towards the periphery. It consists of protophloem and metaphloem. Phloem consists of sieve tubes, companion cells, phloem parenchyma, phloem fibers are absent in the primary phloem. Phloem conducts organic food materials from the leaves to other part of the plant body. So next to phloem we can see cambium, that yellow color layer, no, that is called cambium. Cambium consists of brick-shaped and thin-walled meristematic cells. It is 2 to 3 layers in thickness. These cells are capable of forming new cells during secondary growth. So next to cambium we can see xylem. Xylem consists of xylem fibers, xylem parenchyma, xylem vessels and xylem tracheids. Vessels are thick-walled and arranged in a few rows. Xylem conducts water and minerals from the root to the other part of the plant body. Next, pith. The large central portion of the stem is called pith. It is composed of parenchyma cells with intercellular spaces. The pith is also known as medulla. The pith extends between the vascular bundles. These extension of the pith between the vascular bundles are called primary pith rays or primary medullary rays. The major function of pith is storage. So storage of food, it is a major function of pith. So this is the ground plan and a sector enlarged of transverse section of dicot stem. So the primary structure of dicot stem reveals epidermis, cortex and stele. So it comprises 3 different types of tissue system: epidermal tissue system, ground tissue system. Ground tissue system is differentiated into cortex, endodermis, pericycle and pith, then vascular tissue system. So vascular tissue system with xylem and phloem and also cambium. So this is about the primary structure of dicotyledonous stem. I hope you would have understand the primary structure of dicot stem. That's all about today's class. Thank you.